We'll continue from question number 11, which statements about collagen molecules are correct. So we see both types of secondary structures occur within the molecule. This is wrong because we cannot see anywhere the beta-plated sheet present in the collagen molecules. Large numbers of hydrogen bonds stabilize the molecule. This is correct. And repeated amino acid sequences determine the tertiary structure. This is also correct. So we can say 2 and 3 are the correct options. So D is the correct answer. Moving to question number 12, the protein glutenin gives breadth of its elasticity. The diagram represents the polypeptide glutenin. We can see this is a polypeptide. It has a tightly coiled region over here and it has a loose spiral over here. What describes the structure of glutenin? Quaternary structure because there are both globular and fibrous proteins. No, quaternary structure doesn't mean there are both globular and fibrous proteins. Quaternary structure means that a polypeptide is made up of more than the protein made up of more than one polypeptide. That is, option A is wrong, option B is wrong. Both of them have the quaternary structure here. Now, option C is secondary structure because the loose spiral is an alpha helix. No, that doesn't make it because we can see it here. Tightly coiled region is also here, so it's not only secondary; it has higher than that. So D, the tertiary, because the different regions form a 3D shape. Yeah, we can see the 3D shape is formed here. So D is the correct answer. Now, question number 13, which diagram correctly shows hydrogen bonding between water molecules? The symbol delta indicates the partial charge on an atom in the water molecule. So, first of all, we must know that water is H2O, means there are two atoms of hydrogen attached to one atom of oxygen. They have a covalent bond between them and share a pair of electrons, but oxygen is being more electronegative. It attracts the shared pair of electrons towards itself. So oxygen has a small negative charge in it, whereas hydrogen has the small positive charge on it. So first of all, we can say A is wrong. Sorry, A can be a correct answer because it contains 2 hydrogen, 1 oxygen. C, B, they show it contains 2 oxygen molecules, so this is wrong. D, it shows 2 oxygen molecules, that's wrong. Now we have to select out of A and C. A shows that O has delta positive, H has delta negative. That means that is also wrong. So C is the correct option where O has delta negative and H has delta positive charge on it. Question number 14. Aspirin inhibits an enzyme by reacting with an amino acid that forms an essential part of the 3D structure of the enzyme. Part of the aspirin molecule binds firmly with the amino acid. What describes this inhibition? Competitive inhibition? They didn't mention it, that it binds at the active site. Non-competitive inhibition? It can be that. And a reversible inhibition? It can be only the non-competitive inhibition. Because we don't know whether it's reversible or not. It's not mentioned. Question number 15. The table shows the Mitchell's Menten constant Km for 3 enzymes. Which interpretation of the information is correct? Enzyme C has a Vmax of this. We cannot say from Km value. Enzyme C will reach, we do not say. And the Km is always, Mitchell's Menten constant is always related to the enzyme affinity for the substrate. Lower the Km value, higher is the enzyme affinity for the substrate. So we can see it here that C has the Km value 1.4 10 power negative 2, negative 4 and negative 6. So negative 6 is the lowest Km. So F has the highest, greatest affinity for enzyme for the substrate. That means C is the correct answer. Now question number 16. The diagram is a drawing from an electron micrograph of a typical animal cell. How many layers of phospholipids will a molecule of oxygen cross to travel directly from X outside the cell to Y? So we know each membrane contains 2 phospholipid layers. It means each membrane is a bilayer. It contains 2 phospholipid layers. So we'll see how many membranes the oxygen molecule has to cross from here. First of all, the cell membrane, then the outer membrane of mitochondria, then the inner membrane of mitochondria. So these are 3 membranes. 3 membranes multiplied by 2 because each membrane has 2 phospholipid layers. So 6 phospholipid layers is the correct answer. How could water molecules cross the cell surface membrane of an animal cell? Carrier proteins are usually the ones which can change their shape. We don't use them for the transport of water. Channel proteins, yes, they have the aquaporin channels inside, and cholesterol molecules, they are hydrophobic. So it is 2 is the only correct answer. Now, question number 18. The diagram shows a xylem vessel in a root and 4 adjacent parenchyma cells. As water moves up the xylem vessel, it is replaced by water from the cell D. Water in the cell D is replaced by water in the cell C, resulting in a flow of water from cell A to the xylem. Which cell has the least negative water potential? Least negative means more positive. More positive means higher water potential. So A should have the higher water potential than B, and B then C. So A is having the highest or least negative water potential in this case. So A is the correct answer. So we'll stop here in this video with only these MCQs. We'll continue with the other questions of this worksheet in our